So the first exercise we have been through from the previous class.
Exactly.
Yes. And I want to ask this question, the type of
Right. What is the type of the splicing frame? What are the different types of splicing frame? What could it be?
this will be this will be one of three cases that you introduce us from the class, and we have showed here.
Exactly. You see it, how nicely you show it. It's perfect what you have done there. It's exactly what you need. Can you please, what you have done is absolutely right, and it's the correct thing to have done. Take out the PowerPoint slide that we, or maybe, do you have the PowerPoint slide?
Yes. Shame.
Sorry?
No, I use the Word file to do homework.
I'm not sure I understood the question. Could you please repeat it?
Yeah. Here is the PowerPoint.
Right. Yes. Hold on a moment. So splicing frame 0 is the one that you use the codon before the GT and the codon after the AG, correct?
Yes.
Right. Can you go please back to what you had done in your own Word, in your own Word document, in your own work? This, you have the codon AAG. What does it code for? What is the amino acid that it codes for?
AAG.
Take a genetic code, go to the internet, find the genetic code.
Glycine. AAG.
Right. What is the amino acid that you have got yourself? Do you know how to find this information? If you go further up where you have your sequence, go to the M1 and M2, bit further up, where you have the annotation. There is an annotation for M1 and M2 in your sequence file. Further up, where do you have M1? There you are. What's the translation? What is the last codon that you have there?
Lysine.
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 And what is the codon that you found yourself?
Glycine.
Also lysine. Lysine.
Right. What is the first codon? What is the GTG? What does it code for?
GT.
Do you have a genetic code, or can you get a genetic code on the internet?
It's valine. GTG is valine.
Can you share it with us, a genetic code, so we see how we can... Who has said that?
You can go to Google Chrome.
Do you understand what we are trying to do now? We are trying to see, you already showed us that there is a lysine and that AAG, you said, will be, well done, perfect. So show to us how you figure out what was this that we are looking for? What is the codon that you're looking for? G?
GAG.
GAG. So what does it code for?
GAG.
No, GTG.
No, I think it's a GCG. UG.
G.
Udemy.
Spelling here.
There is G, U, G.
Yeah.
Van.
And what is this?
Berlin.
Can we go back to see what it was? And then you had the license. What was the code? Let me write it down. You say that you have GUG, which is the Valin, and then you had the license, didn't you?
Yes.
What was your code for that?
License is AAG.
AAG, can we see it? And now you go to your Word document.
Here.
You have AAG, which is a license, and then you have GTG, which is GUG basically.
Yeah.
Yes.
Which is a Valin. Is this clear how you find it or not?
Yeah, it's clear.
To everybody?
Yes,
Yes.
all of us.
Yes, it's clear.
Okay, perfect. The remaining exercise, the remaining questions of exercise 1, they are straightforward, correct? There is nothing difficult there.
Yep.
Yes.
Okay, the only thing is, do you remember what is the M1, M2, and the CHS? What are these motifs there? This M1 and M2 that you have nicely colored there, what do they reflect? What do they show us? What do they do, these motifs there?
Like if after the arrangement, the M1 and M2 appear in the final mRNA, the antibody will be anchored in the surface of the B cell. And if in the final arrangement, if it is CHS, the antibody will release into the
Secreted form.
the secret form.
Perfect. Well done. Exactly. This is what we are looking at. If we have the CHS, is the secreted form. If we have the M1 and M2, they're the membrane form of the isotype. Nice. So any other questions on this exercise? No, correct? Everything's all right? Have you understood?
Yes.
Actually, I have
Yes.
Yes, yes,
question.
please.
So I can stop.
about in the exercise 6. No, can you still
Okay. Wait.
about the exercise 6?
So someone is asking a question on which exercise?
She asked about the exercise 6.
Exercise 6. Have you done exercise 4, all of you?
Sorry, my internet is unstable.
Mm-hmm. Okay. What is the question on exercise 6, please?
Actually, it's about the collider period image.
Yes.
Yes. So if we use the access to the request, and in the lab there is a link that when you click on it, it will go you through the decoding here.
Okay.
But if we use the sequence, the picture is different. I still don't know why.
Right. I think I know. I'm guessing basically because I have not seen what exactly you're doing, but I'm guessing. And my guess is that you have taken the sequence without the gaps. There is a precise IMGT numbering, as we said. So most probably, I'm guessing that you took the sequence without the gaps. That is, for instance, you see tryptophan is at position 41, but you see that there are some, it has to be at position 41, cysteine has to be at position 23, therefore there have to be some gaps added in the arrangement of the sequence in order to have the 5 amino acids conserved at their positions, like at 89, 104, and 118, we should have these positions fixed with specific amino acids. In case you have not given the gapped sequence, then the representation will be different. That's what I think. Now, if you want me to look more closely at your results, you should send me the precise sequences that you used. Is this clearer?
Okay, thank you.
And actually, when you use IMGT Quest at the bottom, when we were going through the different outputs, it gives you the sequence in FASTA format, both in gapped according to the IMGT numbering and without gaps. So you should be using the gapped sequence. Gap is with gaps, is with holes, is with some holes in order to respect the 5 conserved amino acids. Okay?
Okay, thank you.
Other questions?
Excuse me, actually the sequence we use have the gap, but it still give the different results. So can I redo our step again so you can see it?
Of course, of course.
Yeah.
You can share your screen and you see, and then we'll have a look at it.
Yeah. Can you see my screen now?
I see your screen now, yes.
Oh, yeah, thank you. Yeah, so at
Well,
first
I see it a bit small, but I see it.
Excuse me, can you see it clearly?
VDJ region amino acid sequence in FASTA format.
Yeah, so we copy this sequence and we paste in the domain gap align here. No. Okay, so firstly, we click on the collier, the point here, the result here.
This
You
is the
see-
easiest that you can do because this will give you certainly the correct result. And then you can look at your sequence. It starts with Q, V, Q, L. Okay, this is the right sequence.
Yeah. But in the case we copy the sequence.
Can I see what you are copying?
Yeah, I copy this sequence. It can have the gap here.
Okay.
Yes. And I paste it in here.
Wait. Domain type, variable, number of layers.
Yes.
Okay.
Yes.
Background color.
Yeah, we choose
Okay.
the first
Sequence. Why do you does it tell you which format? First of all, this does it tell you to give it in FASTA format? Okay, go ahead, show, and then we'll see if it's correct or not.
No, it doesn't have to be the FASTA format. You
Okay.
shouldn't
Exactly. I think you see. Look, hold on. No, wait. Look at
Okay.
what look at the representation. You have the greater than symbol, and then you have the DQ. So basically, this collier de perle took into account the very first thing. This is why you don't have the same thing, because it gave
Okay.
so if you get rid of this one, get rid of the greater than symbol and the accession number where you submit your sequence. No, a bit further up. You cannot change this. There, you can get rid of this blue thing, yes, and then
Okay,
show it again.
Oh, uh, oh,
Now
we
you
still
see?
have a different.
What is the difference now?
I will see CWMC.
In the junction,
Yeah,
like
the
in
junction
the-
is different here. We don't know why. I will divide the screen into two parts for you
Yes.
to easier to observe. Here.
The correct one is the one to the right. Is this for from VQuest?
Yeah, this one I take the link directly, the link, and this one I paste the sequence.
Right. Can I see? Look where the difference comes from. The difference, the only difference there is that at the CDR3 at the bottom, so there is one gap missing. You see at the very top of this violet CDR3, there is SSG, and there has not been introduced a gap, S-gap, SGY. So if you go to the left screen, go to the your left screen, go a bit further up there, and introduce manually a gap in your sequence. After the first S on the CDR3, there is exactly put a gap there, put a dot.
Yeah, I put it here.
Yes,
I show it
show.
again. Okay, they are the same now.
They are the same. So if you go, where did you collect? You collected this sequence from the previous file. Go back to the, right. Can we see where's this SS?
It's here.
Right. So maybe, most probably, there is a little mistake here. There should have been a gap introduced.
Yeah.
You see how we managed to find the little mistake? I don't know why. This can you send this to me, and I will look into how this. Can you send me? What can you send me? Can you send me this sequence? Send me this sequence.
Okay.
In the chat, and I'll see why it was given this mistake. Put it in the chat, please, and I'll look with later on. Now we found out. I'll look with in my team with the computer guys how this can be changed, can be fixed. But this is a little glitch. I don't know where it comes from.
So in conclusion, this link will give us the correct answer, right?
The other will give the correct answer as well. The thing is that there was, they are the same answers. There are no different answers. The difference was that the output here, for some reason, it has not incorporated the gap, one gap. I don't know why the gap was not incorporated there. And because you copied paste this sequence to the Collier de Perle tool, without the gap, it gave you slightly different representation, visualization.
Yeah.
Now, I cannot answer why exactly this gap is missing from your sequence. I have to take more time to look into this. I could come back to you next time to let you know. I'll discuss it, but then I'm not going to do it during our class now. I'm going to do it after the class.
Okay, thank you.
You're welcome. Any other questions, please?
I have a question about the question with chronic lymphocytic leukemia.
Yes.
So I looked over some publication on PubMed, and they said that the mutation rate greater than 2% may be correlate to the disease. So, but there are many documents, and I think the biomarker for that disease is not fully applied yet. So I don't know if it is the right answer or.
Right. Actually, the answer is that you correctly addressed it. So the idea is we would like to have quite a big number of variation in order to be able to attack any antigens that they might be available in our body at any given moment. If we have too limited number of antibodies, that is, if the percentage of mutation is too low, then this is a bad prognosis. This is a bad prognostic. This is bad news for the patient. We would like to have several mutations, and the mutations they coincide. They reflect a healthy individual. They reflect a good, healthy condition. Now the percentage, as you said, in the literature is said somehow 2%. So if you have 98% identity, that is 2% mutation rate, it's not that favorable for the patient. Is this any clearer?
Yes, I think it's clear. Thank you.
Okay. Any other questions on this? All the rest was fine for you? I suppose because we had looked at it together, I suppose it was kind of manageable, right? Right. If there are no any other questions, I prepared a project for you today. This project, I would like, this time, I'm not going to present the project. I have given step by step what we are supposed to do. However, I would like you to present this project. We discussed last time, and I will repeat this time, what is humanization? We said we might have a mouse antibody and we have a human antibody. We found out that the mouse, being infected with a specific antigen, developed an antibody response extremely efficient to this specific antigen. The same disease is found in humans, and we would like to have this mouse antigen. At least we would like to have the efficiency of this mouse antigen. If we were to take the mouse antigen and inject it to humans, most probably nothing would happen, because either it would not be administered properly or it would be aborted. There are two terms that we slightly introduced, and I'm repeating them now, last time. The terms are chimeric antibody and humanized antibody. A chimeric antibody is an antibody that has got constant regions from human, and the antigen binding region is from another species. This is the chimeric one. Chimeric is a Greek word. To me, it is obvious what it is, but maybe not to you. Chimeric is when you have two different things put together. So you have a part of a mouse or a rat sequence and a part of a human sequence. They get together, and this is called a chimeric antibody. Climatic. I'll put it here, the term of it, chimeric. I put it in the chat. And then we have the so-called humanized antibody. The humanized antibody, it consists of constant human region and the antigen binding region. The frameworks, now that you know what is a framework and what is a CDR, the frameworks are human and the CDRs, the complementarity determining regions, or in other words, the three loops that will bind to the antigen, they are from the mouse or the rat species. The advantage of a humanized antibody is that we have much less of a mouse sequence in the total, in the final product, in the total antibody. If we have much less mouse sequence, we have much higher probability that the antibody will not be aborted by our body. Is this clear to everybody? Right. The project that we are going to do today, I ask you to work, you can work 3 together, 4 together, I don't mind. I'll give you like half an hour or something, and you'll come back and present it to us, all of you, is to do in silico. Do you know what is in silico? In silico, it means that you'll do it with the computer. There is in vivo, when you do it in an organism. There is in vitro, when you do it in the lab, when you have the flasks and this and that. And there is in silico, when you do it at the computer. We'll do in silico humanization of a mouse antibody, of an antibody. So basically, we'll start off with a mouse antibody. We'll detect the CDRs. We'll try to find out a human antibody that is equivalent, close to the mouse one. We'll take the human one and we'll graft. The word is to graft. That is, we'll cut with scissors. These are molecular scissors if we do it in vitro. In our case, we'll do copy and paste on a Word document of the CDRs into the human skeleton of the antibody. Is this conceptually clear, what I'm discussing, what I'm describing to you? Right. If it's conceptually clear, I'll be more precise on what I would like you to do. This is the project for today. I'll share my screen. Humanize, okay, it's in French, but it doesn't really matter, this one, because there are links and databases. So this is humanization of a mouse antibody. And then I'm telling you which databases we are going to be using. In any case, we have already used these 2 databases, but it doesn't matter. I repeat them here. You'll be using IMGTV Quest, Domain Gap Align, and Collier de Perl. The very same tools and databases that we have used so far. So here is a synthesis. How can I go further down? These are the steps, all the different steps. All of you, you should at least get up to step 7. It would be great if you go to the very bottom, but at least you should go to step 7. So first of all, I give you a mouse sequence that you can retrieve from this database. Because this database, it's a huge database, and sometimes it's a bit slow over the internet, I have provided in this Word document the mouse sequence. You can extract it from the Word document. This is the accession number. You could go and retrieve it from the database. Then you are going to use IMGTV Quest in order to get the nucleotides and the amino acids of the variable heavy mouse sequence. The antibody has got this Y-shaped. We want to get the variable heavy of the mouse sequence. We're going to visualize it in Collier de Perle. We're going to use IMGT domain GAP-ALI in order to identify the genes and alleles of the human that they are the most closely related to our mouse sequence. And then we're going to do what I told you, this grafting. The grafting is that we are going to take the CDRs from the mouse sequence and the frameworks from the human sequence. In order to delimit, to figure out where's the beginning and where's the end of the IMGT CDR or the IMGT framework, we'll use Hy-Vee Quest. I had already told you how you can do it. You might have forgotten. It does not matter. We'll see it again today. We'll use domain GAP-ALI in order to analyze. Up until here, you will have finished with your humanized sequence. You can do some extra steps. You can use domain GAP-ALI in order to analyze your humanized sequence. You can visualize it with Collier de Perle to see that everything is all right. We'll see a new tool, gene frequency, that you can use in order to figure out how frequently a human gene is expressed. And why is this? If we are using a gene that is extremely rarely expressed, it might not be the best option. We should avoid genes that they are really rarely expressed. And then, an important step, we can use the humanized sequence at the structural level with the hydrogen bonds in order to figure out if there at the mouse sequence, at the mouse antibody, if there are any hydrogen bonds that we are going to break if we don't use the mouse frameworks. It will be much clearer what I'm discussing and describing now when we actually do it. So basically, how do you want to work? I suggest that you have a quick look at this document. I have given you all the answers. However, I would like that we all do it together. So I would like every step, each one of you will do it and will share her screen and will show and explain to the other classmates. I can do the first one, which is the easiest one. So basically, I have gone down to the document and I give you here a mouse sequence that you can retrieve it from this database. Basically, you can take this accession number, you give the accession number to the interface of LigmDB, you wait, eventually you'll retrieve this sequence in FASTA format. If we all of us go to the interface of LigmDB now, it will not work. Or it will take too long time. I don't want to waste our time on this. I would like that you copy and paste this sequence from the file that you have been given and go to the second part. So the second part, I'm asking you to use IMGTV Quest in order to get nucleotided amino acids of the mouse sequence. Now this is a static. I have copied and pasted the result from the online tool to my Word document. I would like you to carry it out on the internet. Even if you have the answer here, it doesn't matter. It will allow you to validate your results. And then I have given you something else as well. I have some difficulty navigating. Here I am. I have used some advanced parameters. I would like you to play around with them. So far, we have not used them. You see, I have said search for insertions and deletions in V region. And then I have said yes. So carry out a VQuest, IMGT VQuest, without this advanced parameter first, and then with this advanced parameter, and observe carefully what is the difference. And then I have given you even the results, the VD region, nucleotide sequencing FASTA format, and in amino acid. So basically, I'm asking all of you to carry out the second step. The first step, I told you, you take the sequence, the mouse sequence. I'm asking you to carry out the second step, which is a simple VQuest. All of you, you should be able to do it. And I'm asking you to do it carefully with two different options. With and without the advanced parameters, see the difference, and then show it to us. Is this okay? Off you go. Please, whenever you're ready. So the thing is, I know that some people will go faster, other people will get a bit lost and this and that. I try. We are from distance as well. I try that we are all in the same boat and we all go together up until the end of this project. So please, I need your help as well. I'll stop sharing. Whenever you are ready, you should kind of speak up. Someone speaks up and share his or her screen to show us the results online. Okay?
Yes, we got it.
Perfect. Go ahead.
So I think I will share the work.
Thank you.
Here. So, so we just translate all the question to English.
We have the second question. Yes, but you are sharing your Word file.
Yes. You need us to share the.
Exactly. That's what I would like you to do
website.
this.
Yes.
Perfect. So what have you done there? Homo sapiens, is this a Homo sapiens sequence, the one that you are giving?
No, it's muscular, no, species.
Exactly. So go back to your there.
The species, yeah, the second.
And this you
first
have not
we choose no have advanced parameter.
with no advanced parameters. Okay, yes.
Oh, okay. So, let me share. Okay.
Okay.
Can you see my screen?
I see your screen, yes.
I will come back to the second step. Just wait a minute. Request. I enter the request. Okay, it's here. I choose the most muscular species, IGA. My sequence is here. Do you want me to choose the advanced parameter or not?
It's up to you. Have you done with both of them?
Okay, so I will choose the advanced parameter. Okay, this is my result.
Okay. So from this sequence, what would you be interested in? Would you be interested in keeping the frameworks or the CDRs?
Yeah.
Exactly, the CDRs. And you see exactly where they are located, correct?
Yes.
Okay. Did you notice any difference when you run it with advanced and when you run it without advanced parameters? And what was this difference?
We have the result here. We can observe it. This one is without parameter. CD1, CD2, and with parameter. Oops. Okay, maybe I will rerun for the option without parameter. No. Start. I will divide my screen into two parts for easier observation. This one is have parameter, and this one is without parameter. CR here, CCZ, D, CR2, AT. We have a difference in this position after the CDR2, I think. I think we have the difference here.
What is exactly the difference? Which is with the parameters, which is with the advanced parameters, and which is without the advanced parameters?
The advanced parameter here is we search for the insertion and deletion in the V region.
Exactly.
Yeah. So we have the deletion.
Right. So basically, this is a sequence. It could be a sequence error. There could be an insertion or deletion. And the program is looking to see whether this is the case and then corrects it in order. Look at the percentage identity you're being given with and without advanced parameters. Do you see the difference? 97%, and then the other is 90%. When there was the correction with the advanced parameters, insertion, deletion, and this was taken into account, the percentage of identity increased a lot.
Yeah. Mm. Okay, I got it. So
I
now
have
we
a
will move
question,
to
Professor.
Okay.
So with the advanced option is better, or we should use the no advanced?
When you should use it, when you see that the percentage, I mean, guys, look, you have a mouse sequence, you compared it against the mouse genome and you found 90% identity. How could it be? Usually we should have much higher percentage identity, because we are comparing it against what? We are comparing it against the genomic variable joining and diversity genes. So if it's an express sequence, if it's an antibody which has got a variable gene, why would it have so low identity score?
Okay, thank you.
Of course, you might answer that there are these somatic hypermutations and there is the junction, but they do not account for 10% of diversity. They account for much less. So basically 97, at least 96%, 97, 98, 99%, is this the scores that we are looking for and that we are expecting. If it's lower, then we start thinking maybe there is some insertion deletion that happened.
Oh, okay.
Is this clear now why I gave you this? This now we go into the advanced parameters of the users of the tool.
Okay.
Okay?
Okay. So now we will move to the next step.
Mm-hmm.
Next step. Này có sai thế.
We move to the third step, exactly. Well done.
Analyze amino acid sequence. Okay. Now I will extract the amino acid sequence. This one.
Okay.
Click here. Yeah, I got the graphical representation.
Perfect. So what basically you need to, okay, you have it. Does it speak to you? Do you understand what you see? You have 5 conserved amino acids. Are they placed at the right positions? Are the amino acids that you should be looking at? Is this a heavy chain or a light chain? And how can you decide which one is which? What are we looking at? From the Collier de Perle, you can give the answer. What are the 5 amino acids that they are the conserved ones, please?
We have C stand for
Okay.
C, WLC and W.
Correct. At the right
Cysteine,
positions.
tryptophan, lysine, cysteine, and tryptophan.
Right. The cysteine at position 104 is not always a cysteine. No, sorry, I said a mistake. At position 89 is not always an L.
Okay.
Right, but it is always this blue color. What is it? Is it hydrophilic or hydrophobic amino acid?
Hydrophobic amino acid.
All right, perfect. So at position 89, there should always be a hydrophobic amino acid. At position 118, there is either a tryptophan, the W, or it's a phenylalanine, the F. Which one is for which chain? Which is for the heavy chain and which one is for the light chain?
Tryptophan is for the heavy chain.
Exactly. Where are the CDRs?
CDI is here.
Mm-hmm. CDR1, 2, and 3.
Yeah.
What are the prolines? Where are the prolines? They are the yellow ones, okay? And they are the small aliphatic amino acids that they give flexibility to the chains.
Yeah.
Correct?
Yes.
Perfect. So,
Yeah.
should you go to number 3 now in order to use the domain gap align? Why are we going to use the domain gap align? What do we use domain gap align for?
Okay, we create the gap in the amino acid sequence.
But
And
say this again, please.
we align our amino acid sequence to find the human sequence has the closest, has highest identity with our mouse sequence, right?
Right, but why? What's the difference between VQuest, IMGT VQuest, and IMGT domain gap align?
I think in the request it is only a line for the same species, but in the domain gap line you can use another species.
Right, actually the main difference is the nucleotides and the amino acids. IMGT VQuest, you can use it with nucleotide sequences, whereas IMGT domain gap align, you can use it with amino acid sequences. So we are using this third step that we do here in order, we visualized the domains in the nucleotide. We want to see the same domains in amino acid sequences. That's the main difference.
Oh, okay.
Okay?
We will come back to the sequence here. Okay, show. I got the result here.
Okay. Can you compare it with the result in the Word document, please?
Yeah.
Do you think it's correct? Do you think there are mistakes? Do you think there are some things that they have changed? And if yes, why not? And why?
CWLC. CWLC. I'm sorry, but can you see my screen clearly?
I can see your screen clearly. Well, I can see the left side clearly. I can see the right side not that clearly, but more or less okay. So this is why I'm asking. So you describe, because it's too small, the line, yes, to the right where you have your cursor, it's a bit too little for me. I don't see clearly which genes you are Oops, now it's huge.
Okay, it's too bad now.
We'll get there. Now it's getting a bit better.
Okay.
So what is the, I don't see, I see most musculus, and then what is there? IG, HV 5-9, that's 1 0 asterisk 0 1. Do you have the same results?
Yeah, it's the same with the answer in the document.
All right, perfect. So this is for you. This is okay. I see it more or less. I see that you have done the right steps. I would like you to be careful and see that you have got the right results. If you don't, then we discuss.
Okay.
Is this okay
Yeah.
for everybody? The other classmates?
It's okay for me.
Repeat, please.
Yeah, it's okay for me.
Okay, perfect. Then I ask you once more to have a look at Collier de Perle. And if you see at number 4 in the Word document, I have even given you below all the explanations.
Oh.
What we just said, the blue boxes, they are hydrophobic, they are the inside, the amino acids hydrophilic, they like the water, they're outside.
Okay.
Okay?
Yeah.
So can we go to step 5?
Yes.
The step 5, you'll use the IMGT domain gap align in order to identify the human sequences the most closely related to your mouse sequence.
Okay.
So basically, you're going to give your mouse sequence, but you're going to select the human sequences. And then try to have a look at the percentage identity and the score that will be given.
Okay.
I would like someone else to share his or her screen to show this to us. You can do it, take 5 minutes to do it yourself. Who is raising his hand? Oh no, I'm doing it. I give you 5 minutes for everybody. I would like everyone to do it. And I'm not in the class, so I cannot know if the others are doing or there are only 2 people doing it. So I would like someone else to share his or her screen in a little while.
Think I can share it.
Sure?
Here is the result from the. So first I copy the sequence, protein sequence here, and choose the species is human. If I dial the list human. Next, the same with our sequence. So is the result and receive it on the website and compare it to I divide into two.
Okay.
Yeah. And you can see it clearly or not?
I see your screen. Sure. Do your other classmates see it? I see your screen. I don't read all the little nucleotides there, but I see where you are. This is fine by me.
Yeah. So as we can see here, if the result from the internet is the same result that you give us before, same here you can see this from the human IgHV3 23.03 here is the same. Can you see my screen is divided into two sides?
Hold on a moment. For the moment, I only see one part. I don't see the two parts.
Oh. Okay.
I don't know the others. Do you see two parts or one part? I only see one part.
Me too. I only see one part.
Okay. So I'll show it again. Oh, yeah. Is this okay? Hello. Hello.
Oui. Yes.
Yes. Now, can you see my screen is divided
Yes.
into
Now I
two?
see the two. Of course. Now I see the two parts.
Yes. So we can see here that the result we receive from the internet is the same with the result that you gave us before. And they also give the alignment process with some concept put in here, and it will same with the result from the internet.
Sorry. Say this. Repeat the last sentence, please.
So we can see that the table here is the same, and also the alignment with the find out the concept genes here is also the same compared to the result from the internet.
Exactly. So this is the same result. So basically you have carried out correctly the exercise, but what does it mean? What is this that we are looking at? What is the information you are interested in from this output?
So we can conclude that the from the human species, the gene and the IGHV3 23.03 is the coldest gene compared to our gene in interest genes.
Okay.
With
So
only
basically,
about with only about 76.5 percentage of identity.
perfect. So you are showing us, you started with the mouse sequence, and you are showing us which is the most closely related human sequence to your mouse sequence, correct?
Yeah.
Exactly. Perfect. So and then you can click on this and show the alignment. I have a question. If you go to this where this is blue, the blue color, if you go a bit to the go to the right, even further to the right of your screen, further more. Yes. Your sequence
Can get it bigger.
right. What is this at the end that it gives you? It gives you this V3 and then it gives you J4 asterisk 1. What does it have the second line underneath?
You mean this one? Or this one? So they compare the allele that they found that have the closest genes to our mouse sequence.
Exactly. So you take the V and the J gene, correct?
Yes.
Thank you. Are the others following? Do you know where we are on the Word document? What is the next step that we would like to have a look at?
We should retrieve the nucleotide sequence for the human IGHV3 allele 2301.
Mm-hmm. So should I let you a bit of time in order to do this? Or do you want to do it while we talk?
We can do it while we're talking for faster.
Okay, go ahead.
So I can give you share your screen. It's okay.
No, you can share your screen. You can do it.
First, you should go to the gene database.
You have a really nice picture on your screen.
You should go to the gene database from the
You can still share your screen.
mine.
Yes.
Okay, so I will just continue to the part of retrieving the sequence. Okay,
Mhm.
not the drafting thing. Ngọc, can you stop sharing because while
Yes,
you're
yes,
sharing,
yes.
I cannot.
Okay.
So can you all see my screen?
Yes.
Okay, so first I will go to IMGT and then go to the gene database. And then I will search for IGH B323. Yeah, and then we see the IGHV23 in Homo sapien. So I will click on this. And here we can see different allele of the gene. And then I will choose the allele 03 with this accession number.
Mhm.
And then we can have the sequence. Okay, we'll. Yeah. That's the sequence. The sequence. Oh, the sequence not the same, I think. There's some difference in the two sequence.
Yeah, it's different.
Okay, I'll try. See? Right. 22. Oh. Oh, this one. But the accession number is not right. Two, 4, 2, 2, 3. VG. So I think the accession number is right, but I don't know why it's a little bit different.
Mhm. The
Like,
difference might be because, hold on a moment. I see this. The difference might be because of the alignment.
the alignment,
Where do you see the difference? I'm trying to see where do you see exactly the difference?
like I see like the beginning is quite the same, but at the end, like
No, actually,
GAA.
the way you should check it is you can take these two sequences, you can
And
copy
align.
and paste and align them. You can use, for instance, Clustal Omega. Go into the internet and find the Clustal Omega tool.
Okay, I will explain. And this one. Value sequence. Oh, it's not in the FASTA format.
Doesn't matter. That's the idea. The idea is you have the answers in the word that I gave you. The idea is that you do it yourself. When you do it yourself, you'll do little mistakes, you'll realize them, and that's the benefit of it. The benefit is that you learn it and then you won't do it again. So don't worry about little mistakes.
Okay, I think it will work right this time. So, like, it has some position that have a different.
Right. To me, it is not exactly the very same sequence. Where you see the asterisks, this is identical nucleotide. The rest is not exactly the same. So, and actually there could be 1 or 2 dots depending, and there are these gaps as well. So it is not the same sequence. However, it's quite similar.
Yes. Oh, that one.
Exactly. Where you have the asterisks is identical.
Yes, this part is identical. So should I try to retrieve exactly this one?
I'm wondering why can you do can you run the exercise again and see the output and see what you chose and see where was listed the sequence that I have put into the Word document? Is this the second one in the list?
Okay, so first I go to GenDB.
I hope I don't give you too much trouble. In any case, you could have used the sequence you chose, but it doesn't matter. Let's work a bit on this if you don't mind. Now that I see that you're interested in it, I want to work with you on this.
Okay, so let me just do
Yes,
it
please.
again.
Yes,
Okay,
I'm here. I'm not moving anywhere.
So as our classmate has done that, so we have this allele has the highest identity.
Look at the second one. What is the second one? What's the percentage identity of the second one?
The second one is
what's the difference? Right. Look at the percent. The first one has got 76.5, right?
Yes. And the second one has 75.5%.
Okay. Right.
So I just choose to go with the one.
The first one, fine.
Yeah, and I will do it again. Okay.
Yes, please.
Okay.
You're at number 5. Let me have a look at my document as well, and I'll realize where this comes from.
Okay.
Because if I open it at my computer, I see it a bit more clearly. It's a bit bigger, of course.
Okay.
So this is the step 5, correct?
Yes, it's step 5.
Right, and you have the very same result, yes, and then you have do you have the same alignment?
Yes, I think we have the same alignment and the same result.
So where's the question then?
Next, I will, like this part, I will try to retrieve the faster sequence of the allele of the IGHV32303 gene.
Right. Yes.
Using the gene database.
Correct. Go to GenDB.
Yeah. So this is GenDB.
Correct.
Can you see my screen? Yes. So I will type in here IGHV3 and
we
3.
are absolutely fine. And then you submit. You choose.
Should I choose the Homo sapien too? Okay. So I will say Homo sapien, submit.
Yes. Correct.
And then I have these 2 genes.
Which is the correct one?
I think the first one.
Of course, it's the first one. Yes.
Okay. So I will go to the gene.
Yes.
And I see many allele here. And the allele we want to retrieve is this one. Like this one.
Right.
So
this is 03. Yes. The 03. Yes.
Is my screen too small? Should I?
I'm juggling between your file and my file, and I see that.
Okay.
That you are retrieving allele 03. Is this the one that you want to retrieve? Why are you retrieving allele 3?
Because it's the allele that has the highest identity.
You are absolutely. Hold on a moment. Yes, you are absolutely right. This is the third allele that you need to retrieve. Correct.
So I will go to a session number AM
Correct.
94. Yes, this is the session number.
Yes.
Wait a moment. Yes, and then
No,
if I
in the download file, you can extract the FASTA file.
You can even
Yeah.
extract from the there.
where? I'm sorry.
No.
Where?
Take on the accession number. Yes.
Yeah, this one.
Take on the accession number. Yes.
Okay,
And
and this one, right?
Yeah. You no. Just take it.
Right? Like this one.
Yes. You can take the.
You are gonna download. Go ahead. You'll see what you're gonna download. Go
Yeah.
ahead.
You have to select the sequence first. Take it on the.
Okay, this
square.
one.
Yeah. And you
Okay.
download it.
You want to download this file?
Yeah. And I give the same sequence with the professor.
Oh, okay. So we'll try. Okay. Oh fuck! It's a zip one.
Of course, it's a big one because it has got many things in there.
I think I would think we should go with the extract.
What? No, can I share the screen and we'll play around and I'll show you. You'll guide me. Can I take over?
Yes, I will stop sharing.
Let me see if I can share.
Goodie.
You'll guide me. I won't be doing it myself. You'll be guiding me. I'm at the IMGT main page, okay? Let me get this out of the way. You see my screen, correct?
Yes.
And then you say that I go to IMGT GNDB. That you know how to do it, correct?
Yes.
You are interested in Homo sapiens?
Yeah. H is here.
Where's the Homo sapiens?
The
It's here, right here.
hetero.
The H number. And the H. Here.
Okay. And then give me the gym name, IGHV
3.
3.
33. Hello?
3.
Dash
23 point. 23.03.
I'll go to my, hold on a moment. I cannot understand what you're saying, the number. So I'll go to my Word document to see what is the number.
So my friend had given the name on the
323.
chat box. Yes.
Are you correct with this one? Are you all right with this one?
Yes.
Yes.
And then this is what you did. You chose this
the first
one.
Yeah.
And then you go to this one. And then you say that this is the right one. And this is the accession number that is given. This is germline DNA. There are several other sequences. You go here, the table which is below, you have the third allele, and what can you retrieve it? You can retrieve the nucleotides of it, you can retrieve the amino acids, and here you can retrieve the V region, or you can retrieve the L part 1 plus the V exon. Maybe you don't fully understand what this is, and first of all, if I click here, I can click here. What do I get? I get a list of all the alleles. This allele here is the third allele in nucleotide sequence. And this is the V region. You see how simple it is? If I go back, say this again.
Yes, we can see it.
If I go here, I can retrieve all the alleles in amino acid sequences. Look, this is the third allele in amino acid sequences, and actually it's a gapped sequence. However, you might have a question. What is the difference between the V region and the L part 1 plus V exon. Does anyone remember? If you don't remember, look at what I do. I click on this logo, the IMGT logo. I'm at the main page. I go to the bottom of the page. I say prototypes, for instance, and I search within the IMGT pages. There you are. I find the page immediately. Do you see my web page?
Yes,
Yes.
we can all see.
Right. This is the V gene. Remember that we had looked at it when we were looking at the genome level. We had the L part 1, we had the L part 2, we had all this information. And now at the table, it was stating that we are looking at the V region. Look, from here, from the beginning of the framework 1 up until the end of the CDR3 is the V region. Depending on what you're interested in downloading, you can choose either the V region or it was telling you the V exon, right? Is it clear how you can download? Can I stop the sharing and can you do how now you should be able to download the sequence yourself? Is it clear
Yes.
or not? Do you want to do it and I can help you?
Dear Professor, I think the sequence that I has downloaded is kind of the same with the sequence you just show us earlier, but it's too different. It's different from the sequence in the Word file. So, like, can you show the show the screen of your screen when you retrieve the sequence of the different allele?
Hold on a moment, I'm sharing my screen again. Is this what you want, right?
No, I think you should go move backward a little bit.
Right. My idea was to show you how you can retrieve the sequence. This is the right sequence for the third allele, but we are saying that this sequence is slightly different to the one in the Word document. Is this what you're saying?
Yes, it is a little bit different.
I think I'll check this. But it doesn't matter. It might be an update in the database. I'll put in my look. Let me share. Now I understand what you're saying. I'm in my Word document, and you are saying that this sequence in the Word document, it is a bit slightly different to the one we just retrieved. Do I understand correctly?
Yes, yes.
Right. This, I did not do it yesterday or today, this file. I will check whether there has been an update of this sequence in the database. So this is not too big of an issue. It might be because this file is from last year. It might be that there has been an update of this allele in the database. I'll check this. Okay?
Yeah.
So now you have your sequence. It doesn't matter if it's not this one. You have your human and you have your mouse sequence. Correct?
Yes.
So now we go to number 7. Actually, I think I might even know what is the difference. We have the human sequence here. '23. Which is the accession number? Is this accession number that you have as well, or do you have a different one?
We have the same accession number.
Same.
Okay. Well, in that case, I don't really know. I'll look into this and I'll get back to you if there has been an update in the database. I have 2 questions to come back to you. I don't really know. It doesn't matter. The sequence we downloaded from the database right now is the one we'll be working with. The idea now is to identify the frameworks from the human sequence and collect just these ones. Do you see my screen? So basically you'll copy the ones that they are in green. Okay? Are you with me?
Yes, we are here.
So how about the step number, what is this? Step number 7, is this clear to you?
So we just extract the frame, right?
Exactly.
Okay.
You just copy the green parts. This is the framework. And if you see further down, you extract from the mouse sequence the CDRs, the yellow parts. And if you see at the bottom of the number 7, I'll share it again. You should have something, you should have this result. You should have the green part, which is the framework 1, CDR1 from the mouse, framework 2 from the human, CDR2, framework 3, CDR3, and this, I explained to you, this is a mouse sequence. This is the framework 4 from the mouse sequence. The mouse sequence was a bit longer than the human sequence. But because they're really close to each other, the mouse and the human sequence, instead of not having anything, we decided to include the framework 4 from the mouse sequence, although our goal is to have frameworks from human sequence. Is this clear? Is there anyone trying to do this cutting and pasting? Or have I lost all my audience?
No, we are still here.
Okay, and are you trying to do this, or do you need any help, or where are we now?
Maybe just give us 5 minutes so we can like figure it
Perfect.
out a little bit. Okay.
Yes.
Thank you.
Yes, perfect. You just tell me when you're ready, please. Any news? Do you need more time or?
Professor, I have a question.
Yes, please.
Yes, but when we use the request to give the V region translation, I am I sure, but is it the CDR3 too large?
You're not sure when you're using the IMGTV quest, you're not sure about what precisely, please?
Can I show my screen? Okay.
Of course.
Okay. So this is the request. Okay, I retrieved the sequence.
This is the mouse sequence that you're using. This
Yes.
E003999 is the mouse sequence. And then
Yes.
you have extracted human sequences. There, yes?
I have extracted the human sequence, the frame. Now I extract the CDR from the mouse, right?
Yes, but no, now you have the human sequences. So you cannot extract the CDR from the mouse. You'll extract, look, the first sequence given
Yes.
is based, the first result is the Homo sapiens result.
Okay, I will thinking more. Thank you.
Hold on a moment. No, stay. You'll understand it.
Okay.
You don't take away your computer screen. You have
Okay.
your mouse sequence, and
Yes.
the first result that you have got is the human sequence.
Yes.
Why don't you go and extract the CDRs, the frameworks from the human sequence?
Now wait a minute. I have extracted the human sequence from here.
Perfect. You have the
Framework?
human, right?
Yeah.
Are you doing it on the nucleotide level, or are you doing it at the amino acid level, the extraction?
The nucleotide.
It's better to do it on the nucleotide level,
Yes.
and now you want the CDRs from the mouse sequence, correct?
Yes.
So take your mouse sequence, the original one that I had given you, this E something, and do an IMGTV quest against the mouse. Do it in front of me now, please. Because this is your last step. You're more or less there. You have this sequence.
Yeah.
You can do it even in
and
the advanced parameters. You do insertion deletions.
Yes.
But if you go further up, I don't see very at the very top of your file here. Go at the very top.
Yeah.
What I don't see, because you have your zoom. What do you choose for species? You can choose whatever you want to. You can choose mouse, mus musculus.
I choose the Homo sapien human.
Exactly. No, you don't choose. If you want the CDRs, choose the mouse sequences.
Mouse sequence. Okay.
Exactly. And start.
Start.
Have you clicked it? I don't know if it's working. Yes, go further down.
Yes.
Yes, go to your alignments. No, stop. If you go further up at the alignment, when it starts the alignment,
Yeah.
yes, what do you see? You see that you have this mouse musculus 5-9-1, the first allele.
Yes.
Correct? And from there, you see the CDR1 at the second line, and you have the nucleotides of CDR1. And this is a mouse sequence.
Oh, okay.
If you go a bit further down, couple lines, stop there, stop. A bit further up. You have the CDR2, but you only see CDR at this line, and 2 IMGT, you see it at the second line. This is your second CDR.
Yeah.
You can copy and paste this, and then further down you have CDR3.
Yeah, but I don't know where the end of the CDR3.
Excellent. Hold on a moment. We'll see it. Go. Perfect. This is a really good question. No, go a bit further up.
Yeah.
Right?
Yes.
You see that your own sequence, the one you submitted, it's a really long sequence. You see all the other alleles that they stop much earlier.
Yes.
So I would stop it at the very, very early. I would not take all the CDR3. I would take what the genes that they have been annotated by IMGT, for instance, the 5-9-1, the 01 asterisk, the asterisk 01, the first allele, goes up until the, look at this, GA. Yes, where does it finish, this one? It has got the CDR3 of this one. Exactly.
Okay. Oh. But where is the frame 4? So you said that we need the frame 4 of the mouse. So I don't understand where the frame 4. CDR4.
There is no CDR4. There is framework.
No, the frame force of the mouse.
Oh,
Yes.
You see?
No. No.
Now you did it with the mouse. You have the CDR3 from the mouse, okay?
Yes. Okay.
Go back and do another IMGT request. And this
Okay.
time you are going to choose Homo sapiens.
Homo sapiens.
Okay?
Yeah. Professor.
You have your mouse sequence, and
Yes.
then you see the Homo sapiens. Once more, you see up until where the CDR3 goes, okay?
Yes. Yeah. Yeah.
Perfect. So you can take this.
Okay.
But this is from your original sequence.
Mhm.
This is the CDR3. If you have done up until there, it's absolutely fine because you have the CDR from the mouse sequence, your own sequence, and then you
Yeah.
have the framework from the human sequence. You put them together, forget about framework 4 for the moment, you have your sequence. You graft them together, you put them together, you have your humanized antibody.
Okay. I will try.
I think you all need to play a bit more. At least 3 people, they followed. The rest, I have no idea if you followed or not. At least three classmates, they followed the project. I suggest that you go through the Word document a bit more. You ask questions. You sit by yourself with your own pace. And then we come back tomorrow and we ask more questions if you have got.
Yeah, but basically, when we finish with the design humanized antibody, we can produce it and validate it by in vitro, in vivo experiment and make money, right?
Exactly, this is correct. We did not go up until the end of the document. If you want to go a bit further, what I suggest, we have done the 7.
Yeah.
And then you have your humanized antibody. I suggest the following. You take your humanized antibody, you can copy and paste it from the Word document,
Yes.
and go into IMGT 3D structure DB. I'll help you. Share your screen.
Okay. Can you see my screen?
No, now I see it. Yes, copy and paste this sequence. Take this sequence.
Yeah.
You can get rid of the spaces, but it doesn't matter. Okay, you have your sequence, correct? Go to the main page of IMGT. You click on the logo.
Yes.
Go to the databases and choose this IMGT 3D structure. No, a bit further down, a bit further down. Your cursor a bit further down. IMGT 3D structure DB, not the tools, the databases.
Oh.
Up to the left. Where you have your cursor.
Database.
In the databases.
Database ở trên này ơi. Ở trên cái tung
Vậy
đi.
hả?
Trên nữa.
Ở
To the
đây.
left.
Đấy, đây, đấy. Đấy.
Okay.
No, GDB, no. The one that's the
Yeah.
one. Perfect.
Okay.
Right? This is a
Yes.
new interface. Okay.
Yeah.
Further down. Search using structural query.
Okay, I paste this here.
No, search using alignments because you don't have structural query. But this, you have to clean up a bit your sequence. If you go search using alignments, not this block, the one below.
Oh, okay.
Exactly. However, look at what you have got there. You need to have your sequence in FASTA form perfect. Get rid of this. Well done. You did not get rid of framework 2 further above?
Oh, here.
Yes. Well done.
Okay, I think I clean up the sequence.
Yes. Okay.
I click shift.
Maybe you should have got rid of some spaces and return keys, but anyway, let's go search and we'll see what comes out.
Yes.
Actually, it was quite nice. Look at this. If you
This
go to
one?
the top,
The top, yes.
this is a summary. What does it give you? I don't see your summary because you have your zoom in front of the top. You put your zoom to the right, please.
Here?
No, the zoom, where we see ourselves.
Ah.
It is, move this out of the way, please.
I don't know how to hide.
You just click on it and you take it to the right of the screen and it will be vertical instead of being horizontal. No, you don't click on your window, you click on the zoom.
Okay.
Anyway, perfect. This is fine. Look at what it tells you. It gives you here, do you understand what is this 7USLB, 7USL, integrin beta 2 Homo sapiens? So this is, these are structures extracted from the protein database.
Okay.
And we could look the structure of this one, which is 5, and the alignment with your own humanized sequence. So basically, what would be of interest, it would be to take the structure and visualize it with the collier de perle.
Oh. But the percent of identity just is quite low, I think.
It is quite low.
Yeah. But it is the best score.
I see to start with, I see that we have not found the same structure, and I'm wondering why. And I would, while we are talking, I'm doing exactly what you did to figure out why we have different outcome. Because you see, I don't have the same structure as you have, and I would like to check it because yours does not seem correct to me, and I don't know why. Whereas mine is the correct one, and I would like to show it to you. So hold on a moment.
Yeah.
I do exactly the same. Oh, right. Oh, I see already where's the problem. Can you share your screen again? Go to the top of your screen. Very, very top, even before that. Look at this. Sequence name 1, you gave your sequence name, and then it tells you amino acid sequence. You have given nucleotides, and it has taken only a tiny little bit because there were a lot of spaces. You need to take
the amino acid sequence.
Exactly. Well done. You need to take your amino acid sequence where you find it at number 7.
Yeah.
Most of the times, if we are extremely careful, we'll find the solution by looking at the output files.
Yeah. Okay, now we had hierarchical identity.
This is why the percentage was not that good.
Yeah.
Once more, you'll have the issues, I think, because you need to take without spaces. So you need to get rid of your gaps.
Oh, I have to remove the gap.
You have to remove the gaps in order to search.
Okay.
If you don't remove, you'll realize the mistake when you see the output. But in order to prevent it, I told you. And then you'll see how much better the result will be.
Yeah, it's nearly 90.
Right? You see it?
Yeah.
Right. Can you take this?
This one?
Take. Now you have, no, this is the alignment. This, you see that the percentage of identity is quite good. Okay?
Yes.
If you could take your nucleotide humanized sequence.
Yes, it's here.
I'm doing the same, actually, what I'm asking you to do. I'm doing it. I'm repeating it with you. And repeat an IMZT request. Now you should know how to do it. I say Homo sapiens. I make immunoglobulin. I give the first day of my sequence. I start. I let it run, and I come to see what you are doing.
You want me to process the request again?
Yes, I would like you to do a request of your own sequence.
Oh, with the humanized antibody sequence, right?
Correct.
Okay. Stop. Okay, I have the result here.
Let me see. Okay. Fine. So you have 96% of identity, correct?
Yes.
And if you go, you see the frameworks, you see the CDRs. Do you see at the alignment that the CDRs is where you have a bit more difference? Why?
Because we use the mouse.
Exactly. Perfect. This is exactly what you're saying. We see here clearly that most probably we have done the right grafting.
Okay.
Okay.
Yeah.
So this is like a little verification, a little validation of your final result.
Yeah.
Do you want to do a very last thing or are you tired?
Maybe we can stop here.
Just do
I
the last
exhausted you.
one.
Okay, fair enough. So basically, I realized that there were not many people who were following today. So I have a question. Are you ready? How have you prepared your monoclonal antibody presentations or not? Is this worth doing a class tomorrow? Are you gonna come? I have another project equivalent to the one today's project. Are you interested in it? Are you not? Should we hold the class? Should we not hold the class? Instead of the project, would you like to present your monoclonal antibody mechanism of action? Should we not have a class at all? And we meet on the 21st and you have your presentations? What do you prefer?
We haven't we haven't done the mono antibody presentation yet. So, classmate, do you want to continue another project tomorrow or something? But one certain thing is we cannot do the presentation tomorrow.
Right. If you have not done your presentation yet, you could take advantage of this time slot tomorrow. I could be with you and you prepare your presentations because you need some time. Instead of doing another project where I'll be talking with 2 or 3 of you and I'll be missing the rest of you, at least I can be present with all of you. But the idea is that you work on your project for the 21st of December. What do you... Yes, please express yourselves.
Yes, I think it's okay.
However, I don't want to tell you you do it on your own like this. At least if you have questions, you ask me, please. So I don't send you a new project. I'll be staying here correcting your homeworks. You'll be working on your mechanism of action monoclonal antibody project. And in case you have questions, you'll be asking me. You'll be asking them to me. Is this okay?
Yes.
Okay.
Yes.
So the plan is tomorrow you will be with us.
Say this again.
That the plan is that tomorrow you will stay with us, and we can have time to prepare the monoclonal antibodies. And if we have any questions, we can ask you. And
right.
Yes.
And then on the 21st, I expect everyone to be ready to start your presentation.
Okay.
Fine. Thank you.
Okay.
See you tomorrow. But the idea is that you're going to be working tomorrow.
Yes.
Okay.
See you tomorrow.
Take care. Bye-bye.
Bye-bye.
Bye.